Today's topic is photosynthesis. You must be knowing the most conventional definition of the photosynthesis is that the green parts of the plant, in presence of sunlight, utilizes water and carbon dioxide as a raw material and produces the carbohydrate. That is the glucose, and oxygen is evolved. Now, if you ask the chemistry of the photosynthesis, you can say it is an oxidation reduction reaction where water is oxidized and the CO2 is reduced. Now look at the systematic definition of the photosynthesis. Photosynthesis is an anabolic, reductive and endergonic process which uses the light energy in order to photochemically reduce CO2 into carbohydrate, and oxygen is a byproduct of this process. And the overall equation of the photosynthesis is that 6CO2, 12H2O, light plus chlorophyll gives you carbohydrate, water, and oxygen is evolved. You must be knowing that the green plants uses only 1% of the total energy and to fix the 2 billion tons of CO2 annually. Now, as I told you that chlorophyll, which is present in the organelle chloroplast, plays a very important role. So now let us discuss the structure of the chloroplast. You know that chloroplast regarded as the food production centers and is one of the largest cell organelle of the mesophyll cells. It's a double membrane organelle and it is surrounded by a selectively double membrane permeable membrane. The matrix of the chloroplast is a stroma, which is rich in enzymes, which is involved in CO2 fixation. The pioneer to work out the structure of the chloroplast was Willstatter and Stoll. Each chloroplast has 40 to 60 grana, and embedded in the stroma are the thylakoids, that is the stacks of the thylakoid, which is referred as granum, and you have 40 to 60 grana in a chloroplast. The region where one thylakoid is in contact with the other is called as appressed region, and all the grana are linked by fret membranes or stroma lamellae or stroma thylakoid, and the end membranes of the grana and the grana margins, which are exposed to stroma, are called non-appressed region. There is a division of labor in the chloroplast. Do you know that the grana or the grana lamellae is concerned with the light reaction and the stroma lamellae with the dark reaction? The thylakoid membranes have or consists of two structurally distinct photosynthetic units, which you call it as photosystem 1 and photosystem 2, the details of which we will be taking up afterwards. The chloroplast has the pigments called chlorophyll and carotenoids. The chlorophylls are responsible for harvesting the light energy. And you know that the chlorophyll is which is present in all the photosynthetic organisms, except the photosynthetic bacteria. B is restricted to higher plants and green algae. Look at the formula of the chlorophyll A, that is C55S72O5N4Mg. Chlorophyll B, here you have C55S70O6N4Mg. The difference between the two chlorophyll is that the chlorophyll A has at third carbon the methyl group, and this methyl group is replaced by an aldehyde group in chlorophyll B. Besides chlorophyll, you have a necessary pigment which you refer it as carotenoids, and these carotenoids are divided into two categories, that is the carotenes, which are made up of hydrocarbons, and of course xanthophyll, which besides having carbon and hydrogen, they have oxygen. Carotene are C40 and H56, and the common carotene are lycopene and beta carotene. Xanthophyll, besides having carbon and hydrogen, they have oxygen also, that is C40 H56O2, and the common xanthophyll includes lutein and zeaxanthin. As I told you that Willstatter and Stoll gave the detailed structure and chemical composition of the chloroplast. Park and Begin, they in 1961 discovered the distinct photosynthetic units, which is referred as quantosome, and they contain about 230 chlorophyll molecule. Now here I would like to add that the biosynthesis of chlorophyll and hemoglobin starts from the raw material succinyl coenzyme A plus glycine, and the biosynthetic pathway of the two are common up to an intermediate compound which is called as protoporphyrin. From here the two pathways are different because, as you know, that in hemoglobin you have the iron, and of course in chlorophyll you have the magnesium. The immediate precursor of the chlorophyll is chlorophyllite. Now we move to the mechanism of photosynthesis. You know that the presence of two phases in the mechanism of photosynthesis was established by Blackman in 1905. The first is referred as light reaction or Hill reaction, and it was R. Hill in 1937 for the first time exposed the isolated chloroplast to light in presence of certain substances which were having the ability to accept the hydrogen, and he was able to evolve the oxygen. The substances which were used by Hill are now referred as Hill oxidants. Now look at the overall equation of the light reaction. Water splits up into H and OH. A is the Hill oxidant, and 2A plus 4H is reduced to 2AH2, and the 4OH gives you water and oxygen. To sum up the equation, we have 2A plus 4H2O, 2AH2 plus 2 water and oxygen. Similar experiments were